Applications of biotechnology. What is biotechnology? Biotechnology is the use of biology to develop new products, methods, and organisms intended to improve human health and society. There are five branches of biotechnology: medical, environmental, industrial, animal, and plant. GMO, DNA fingerprinting, gene therapy are the applications we are going to discuss. GMO. Genetically modified organisms are produced using scientific methods that include recombinant DNA technology and reproductive cloning. In reproductive cloning, a nucleus is extracted from a cell of the individual to be cloned and is inserted into the enucleated cytoplasm of the host egg. The process results in the generation of an offspring that is genetically identical to the donor individual. The first animal produced by means of this cloning technique with the nucleus from an adult donor cell was a sheep named Dolly, born in 1996. Since then, a number of other animals, including pigs, horses, and dogs, have been generated by reproductive cloning technology. Recombinant DNA technology, on the other hand, involves the insertion of one or more individual genes from an organism of one species into the DNA of another. Cornstarch, corn syrup, canola oil, potatoes, apples, pink pineapples are some of GMO products which are available. DNA fingerprinting. DNA fingerprinting is a technique that simultaneously detects a lot of mini satellites in the genome to produce a pattern unique to an individual. This is the DNA fingerprint. The probability of having two people with the same DNA fingerprint that are not identical twins is very small. Just like your actual fingerprint, your DNA fingerprint is something you are born with. It is unique to you. Gene therapy. Gene therapy works by altering the genetic code to recover the functions of critical proteins. Proteins are the workhorses of the cell and the structural basis of the body's tissues. The instructions for making proteins are carried in a person's genetic code, and variants in this code can impact the production or function of proteins that may be critical to how the body works. There are two different types of gene therapy, depending on which types of cells are treated. Somatic gene therapy, transfer of section of DNA to any cell of the body that doesn't produce sperm or eggs. Germline gene therapy, transfer of a section of DNA to cells that produce egg or sperm. DNA therapy techniques. Gene augmentation therapy, gene inhibition therapy, killing of specific cells. This is the end of the presentation. Thank you.